Okay, so welcome everybody. Thank you for attending today. My name is Luis Alvarez, and I'm from the Molecular Imaging Center at UC Berkeley, and I'm one of the co-organizers for the Fluorescence and Advanced Imaging Research, or FLAIR, workshop. Today, you'll be hearing from one of our fantastic sponsors, IBIM Technology. They'll be joining us in person at UC Irvine on January 12th through 15th. Registration is still open for the event, so we hope to see you there. Presenting with us today is Dr. Pilhan Kim, CEO of IBM Technology and professor at the Korea Advanced Institute of Science and Technology in Daejeon. Dr. Kim is a leading expert in vivo cellular imaging with electrical engineering training at Seoul National University and postdoctoral experience at Harvard Medical School. His work focuses on advanced intravital confocal and two-photon microscopy for real-time visualization of disease mechanisms and therapeutic development. So without further ado, welcome Dr. Kim.
Okay, all right. Thank you. Thank you very much. Okay, so first let me share my screen. All right. Okay. I hope everybody can see my screen. All right. Okay, so let me begin. So as Luis just introduced, today I will talk about the real-time intravital microscopy for in vivo, live cell and drug imaging, live animal model in vivo. Okay, so many of you already heard about what is intravital microscopy. But just to give you one introductions. So this intravital microscopy is a technique that can provide this kind of microscale, single cell level images and video from the live animal model. So compared to other imaging modality, it is clear by using intravital microscopy, you can distinguish individual, cells, movement, and their dynamics, and also their interaction in their in vivo in their in vivo microenvironment, as you can see in these images, right? So in this particular example, we are looking at the blood flow in green fluorescence. So you can nicely see this blood flow in the lung of a live and excised mouse model in vivo, okay? And then in that color, we are also looking at the neutrophil. And then you can see, how dynamic they are inside the blood vessel, and then how they are interacting with each other, and then making a small cluster in here. And eventually they disturb the, microcirculation in this site inside the lung of a live mouse model of acute lung injuries. Okay, so this is one good example what you can expect to obtain by using intravital microscopy. Okay, next. And also, not just the cell. So we can also image the, draw, live animal model with process labeling. So in this particular example, we are looking at the liver, live and sized mouse model. And then we inject presently labeled nanoparticle through the tail vein intravenously. And then in the liver, we can see how these, nanoparticle arrives in the liver sinusoid and how they accumulate and distributed in real time. Okay, so this is one another good example how we can use intravital microscopy to visualize the drug delivery and therapeutics delivery in a live animal model in vivo in their internal organ and tissues. So in short, intravital microscopy is a very useful technique that it can acquire these live cellular level images and videos in a live in vivo animal model with molecular and functional informations. So by using our intravital microscope, we have been able to image many organs and tissue, almost every organ and tissue, as you can see in this slide. Okay? So all these images were obtained from the live and it's the live mouse model in vivo. So we have been able to image bone marrow, brain. This is a brain tumor, orthotopic brain tumor, retina, skin, hair follicle inside the skin, and also various tumor model. This particular example is a colorectal tumor model, pancreas, spleen, GI tract, adipose tissue, prostate, placenta in a pregnant mouse model, various lymph nodes, memory tissue, cell transplant. These green things are the transplanted cell in the kidney capsule, kidney, muscle, liver, heart, lung, thyroid, and so on. So actually this list is keep increasing. Okay, so let me briefly, talk about our microscope system. So it looks like this one. And then every optics is integrated into this single box, into, all ordinary packages. And it has quite small size, like 63 centimeter, 89 centimeter in width and length, and 49 centimeter in height. So it's quite compact. So you can easily put this microscope anywhere in the lab or inside animal facility. Or alternatively, we can also use conventional through the optical port via box for the two-photon imagings. Okay. So let me just shoot few example of the four color two-photon imaging data we obtained from the live mouse model. Okay, so I hope everybody can see my screen and follows me. So this is a live image and videos obtained from the skin of live exercise mouse model. So this is a four color imaging data, and this four color was simultaneously obtained with G-stack imaging sequence. Okay, so in this example, we use H2B GFP and then ROSA 26 MTMG mouse model. So we can nicely see P in blue color, which is a pseudo color. In addition to that, finally, in white color, using SHG signal, we can nicely visualize this thermal collagen in the skins. So this is one very nice example. We can obtain, sub-cellular resolution images in G-stack in the any tissue. In this example, it's skin in the live mouse model. Okay, another example. This is a muscle. So in the muscle, we can also very nicely see this myofiber and the multinucleated myofibers. We can also automatically, image the multiple sites and generate these large area mosaic images. And nicely see the arsenite cell, individual arsenite cells, and again, wide area images. So one G-stack images. And based on this real-time imaging capability, we built in the automatic live tissue motion compensation functions. So, as you can easily imagine, so if you image the internal organ in a live animal model, we always suffer, this kind of, tissue motion in microscopic scale, okay, because of the breathing, of the air breathing and also heart beating. So, whenever we image the internal organ, we often see this kind of, tissue movement because of that, okay. But because we have this video rate, real time imaging capability, we can directly track the tissue movement in real time. So if you look at here, this cell, by just looking at this cell and other cells, we can see how these individual tissues and moves around in frame by frame. Okay. Okay. So we just recorded this real-time video and then do the frame by frame, motion compensation like this. Okay, frame 1, 2, 3, 4, then we compensate it and generate the, stabilize the images. So this is the result, the comparison with the motion compensation and without motion compensations. So from this kind of a moving tissue, we can very nicely generate this high resolution, high SNR, very clean images out of this moving tissue. So if your microscope is slow, like 1 frame per second or 2 frames per second, then you will always suffer this kind of image blurring due to these tissue motions. Okay. And then also, we built in this function to be automatic. So this is the UI of our microscope. Okay, so what you need to do is just turn on this motion compensation functions and then press one button. And then as you can see, in just a couple of seconds, it automatically generated these compensated images from the moving tissues. So it's quite useful when you try to image the internal organ in a live animal model in real time. Okay, and then I like to highlight another things. So as I told you, we are capable of real-time imaging with a speed of at least 50 frames per second. And then so basically we can record this high-speed video from the live animal model. And then recently, about 2 years ago, at 2023, we published this one paper about the novel AI denoising algorithm, which is very much optimized for the high-speed video. So not to losing the temporal resolution from this high-resolution microscopic image videos. And then another good thing is it doesn't need pre-training. So no pre-training is required and it's unsupervised method. So in principle, we can implement this algorithm to be automatic. And then we use blind spot algorithm to achieve the denoisings. So anyway, let me jump into the real result. Okay. So on the right hand side, this is the raw video, the raw real time, the real time, raw real time video obtained from the skin. Okay. Again, we use H2B-GFP and Rosa 26 MTMG mouse model, so we can nicely see the nuclei and the cell membrane. So these are the capillary and the hair follicle. And at the same time, we inject the fluorescent red blood cell. So this blue color is actually flowing red blood cell inside the capillary. So we have a very high spatial, temporal, resolution in this video, although we have some noises here. But after applying this denoising, AI denoising algorithm, we can very nicely remove the noise from this real-time video, as you can see in here. Okay, so we can improve SNR more than hundreds times. So another example. So this is another data obtained, another video obtained from the muscle. So we have this, larger vessel here. Okay, and then we can see many, flowing red blood cells here. So we have, very high, temporal dynamics in here inside. But again, after applying this AI denoising algorithm, we can very nicely remove all the noises, and then we can even distinguish these, sarcomere structures inside the myofilament in submicrometer resolutions, and also we can preserve this, very high temporal resolution enough to distinguish the individual, flowing red blood cells. Another example. So it's a little bit more noisy in here. It's another real-time video obtained from the kidney. So we can see this growth structure of the tubule, right? But after denoising, we can also, distinguish this individual, epithelial cells here inside the tubule, right? And then finally, this is another data obtained from the brain, and then this is very noisy. And then our SNR is, very low, like 1.2 or 1.1 even, right? But after denoising, we can very nicely, uncover this hidden signal from this very noisy area. If you look at here, we can nicely distinguish the individual neurons in here. Okay. So our AI denoising algorithm works perfectly with this real-time video we can obtain from the live animal model. Okay. And then the first example I showed you was the nanoparticle delivery and distribution in the liver. And then actually that data was also denoised data. So on the right hand side is the raw data. And on the left hand side is the denoised data. If we zoom in, so and then after denoising, we can easily, distinguish even individual, nanoparticle with a size of 200 nanometer, and then how they are flowing and then distributing inside the liver sinusoid, as you can see in this example. So this AI denoising algorithm is here, really perfect match with our real-time, intravital microscope, intravital two-photon microscope. Okay, and then I like to say another things. Although our microscope is capable of 50 frames per second as a default speed, and this speed is only limited by the number of pixels in y-axis only. Okay, so in other words, if you reduce this pixel number in y-axis, like to 256, then our imaging speed will be doubled, like to 100 frames per second, right? And then it doesn't really matter how many pixels you have in x-axis. So we can take this kind of rectangular shaped frames with even higher imaging speed, like 100 frames per second, 200, 400, even 800 frames per second. Okay. All right, and this AI denoising algorithm is already released, and then this is a full automatic software. So what you have to do is just upload your videos and press a single button. And then after several, after some time, you will, get your, denoised algorithm, denoised videos. And then this software support all these, different video format. So if you have some noisy video you want to try to denoise, then you can easily ask us to try this software. All right. So from now on, let me quickly show, how we image all these different organs. So first, let me begin with the lung, which I showed in the beginning. So in case of the lung, we need still, some additional, device, which we call, stabilized lung imaging windows. So as you can see, the lung is always moving because, mouse have to breathe air, right? So we build up this negative air pressurized imaging chamber here. So we have a small hole here, so we can suck out the air, build up the very gentle negative air pressure inside, which is enough to stabilize the surface of this lung to this transparent cover glass, as you can see in here. Okay. Right, now it's stabilized. And after this stabilization, we further apply our motion compensation algorithm, motion compensation functions. Then we can generate this, sub-resolution, time-lapse images showing the, showing the clustering of the neutrophil inside the vessel in the acute lung injury model. So like this, in just a couple of minutes, we can see this, maybe 7 neutrophil make a cluster and then block the vessel and then disturb the, microcirculation. So no microcirculation in this area anymore, right? All right, so let me quickly show you one single video to how we do this. So we first need to intubate our mouse model, right? And then we connect it to the mechanical ventilator to mouse to breathe air during our little surgery to expose the lung surface. And then we stabilize the lung surface as you can see here. And then we place our mouse model into the our microscope. And then, but actually all this inside is actually heated. This stage is heated, and then this glass is also glass, and the windows can be heated. So everything is heated to keep the mouse body temperature to be stable during our intravital imaging experiment. Okay, and then we can also use the same principle even for the heart imaging as well. Okay, so in this example, so as you can see, this is the heart surface stabilized. So this is 2 millimeter diameter. So this beating heart surface can be stabilized by using same principle of negative air pressurizations. And after that, we can directly see the heart beating and these flowing cells inside the coronary vessel in the heart surface. Okay. And then let me tell you, how long can we do this kind of, interventional imagings? So the answer is as much as we want. As much as we want, as long as we, keep the mouse to be anesthetized. So we can easily, image the mouse to several hours, with injectable, anesthetics. Okay, so but anyway, so this is one example. Obtained the 2-hour time-lapse videos obtained from the pumped little lymph node of this NSIG mouse model. So in as you can see here, so we catheterize the tail vein. So we inject some lymphocytes, T cell and B cell, into the tail vein intravenously, and then observe this how this intravenously injected T cell and B cell extravasating from the high nasal venule in the middle in the lymph node. So this white arrow, these arrows, pointing out the extravasation point of T cell and B cell. Okay. So if you look at carefully, you can see the whole procedure, whole process of this T cell and B cell extravasation. So you get this green one, it comes up right here. And then later, this red one, follow the same way. So it's not like, just a single direction way. So it's more like, attached to the, endothelium and then go into the epicyst subchannel and then get out. So this is one good example. So we can see the cell trafficking in the live tissue for several hours by using intravital microscope. And then the real beauty of intravital microscopy is longitudinal imaging. several days to even several weeks. So we can, after each imaging session, we don't need to sacrifice the animal model. So we can we can recover our animal model, mouse model, and then put it into the cages. And then we can bring the same mice again, do the intravital imaging again, and then find the exact same locations and see what's going on there for several days and even weeks. So this is one example. So every day, we label the blood vessel by using anti-CD31 antibody probe conjugate, vascular sinus in blue colors. Okay, so this is 10 days longitudinal imaging to see the behavior of this transplanted bone marrow cell inside the bone marrow. Okay, so as you can see, at day 3, we can see just few cells here, and they, rapidly proliferate. Day 4, day 5, 6, 7, right? And then suddenly they mobilized and disappear from the bone marrow, and they went out to the periphery. But interestingly, if you look at here, there's a surrounding niche inside these bone marrows. They now have proliferations, transplanted bone marrows and proliferations. So this data, suggests, at the end, it looks like this one. So we can see the whole process, how these transplanted bone marrow cells, fills up the, fills up the recipient bone marrows one by one, right? First in the middle and this area, right? So it's like, sequential, sequential filling up into the bone marrow by the transplanted bone marrow cells. So this is one good example what you can obtain by longitudinal repetitive intraretinal imaging to track this cellular level process in the live animal model. Of course, at any time point we can further zoom in, and then see distinguish individual cells, and then to observe their behavior. So here you can see the cell proliferations, and also in the cell movement here recruited into these niche areas. Okay, so another example. Let me just go through. So this is the again 3 weeks observation of the development of the lipid droplet in the liver. Okay, so this is the longitudinal interventional imaging of liver non-alcoholic fatty liver disease modeling. So in yellow color, we can very clearly see the accumulation of the lipid droplet inside the hepatocyte. Okay, and then let me talk about how we can use intravital imaging for the drug development. Okay, so so as you can as some of you already know, for typically for to develop the drug, it first starts with the target validations, right? And then, as I showed you in several examples, intravital imaging is a useful tool for basic profiling of the target in vivo. So we can, validate the target using intravital imaging in the live animal model, and then to provide the, probable concept of those target. And then, after this hit identification, lead identification, and lead optimization, we can have, several candidates. And then before going into the clinical trial, we normally do, we need to profile these candidate drugs in a live animal model in preclinical testing. And then also here, intermittent imaging is a very useful tool to monitor the drug delivery and also efficacy. Okay. So basically, we can do in vivo, PK, PD imaging analysis by using intermittent microscope. So for that purpose, we have several imaging windows techniques for longitudinal imagings. So first, let me show you the result with the dosage skin point chamber. This is a very well established technique. Okay, so to save time, let me just jump into the result. So basically, this dosage skin point chamber, it looks like this. And then this one can be permanently implanted into the dorsal skin of a live mouse model. And then you can see the surgery procedure from this YouTube link. And then, okay, so after this implantation, this mouse will carry this dosage point chamber for 3 to 4 weeks. And then during those 3 to 4 weeks, we can repeatedly image the, tumor development and the drug treatment inside of these imaging windows. So this is one example. So in here, we inject this human triple negative breast cancer cell line and then B231 GFP. Okay, and then we let these tumor cells, grow for 2 weeks. So we can see this nice, development of this tumor, breast tumor, in single cellular level. And then we label the blood vessel using antibody, again, anti-CD31 antibody, and then we intravenously inject the nanoparticle in that fluorescently labeled nanoparticle. So let me remove the green color. Okay, so we can nicely see how this nanoparticle is delivered to the tumor tissue. At 2 hours, well, we don't see many. At 6 hours, now we see all of the nanoparticle, but they are mostly inside the vessel, as you can see in here and also here. Let me zoom in. But finally, at 24 hours, we can see that all of the nanoparticle are, get out from the vessel, right? 2 hours, 6 hours, 24 hours, and then finally, delivered to the tumor cells. Okay. So this is one very good example to monitor the drug delivery to the target cell, target tumor cell in these cases. We can also image other drug. We monitor the exosome delivery, therapeutic exosome deliveries. So this red color is the exosome, and green color is our target cell in the liver. Okay. So this is before injections, and this is 10 minutes after the injection, IV injection of this exosomes. Okay, and then we can see very rapid targeting of this target cell by the intravenously injected exosome, as you can see in here. At 30 minutes after the IV injection, we can now clearly see the internalizations of this exosome into the cell. So this is another good example how we can use intravital microscope for the monitoring of the drug delivery to the target cell in the target organ. Okay, and then so let me recap this data. So I already show you, explained to you this neutrophil is the real major cause of the microcirculation disturbance in this lung to lung injury model. So this neutrophil can be our target. So to make a long story short, we screen the several potential target, and they find out through this one drugs called abciximab. So, uh, and here, this red color is actually the flowing red blood cells. So basically, this is the data showing the microcirculation in the lung. Okay, and if you look at here, there's no RBCs here. So it means this microcirculation is disturbed in this particular area. Okay, and after treating these drugs, you can see the recovery of this microcirculation, right? So we can see the RBC flows were recovered after this drug treatment, right? So this is the one good example that we can directly, monitor the efficacy of the drugs in the live animal model in vivo before and after the treatment. And then also it's correlated with the physiological, functional restorations, right? So this is the after treatment, and this is without treatment, and this is sham controls. Okay, so after this drug treatment, this physiological function is also restored, and also we can see how they did it. So we target the neutrophil and recover the microcirculations. So this is the whole story of the in vivo validations, MMA validation of the drugs in this live animal model. Okay, and let me jump into the other models, the other technique, which we call abdominal imaging window. So it's basically ring-shaped window. Again, it can be permanently implanted on the abdomen of the live mouse model. And through this imaging window, we can image the abdominal organs, such as liver, kidney, pancreas, spleen, and so on. So let me show you one example. So in this example, we implanted this window to see the pancreas. This pinkish area, pinkish tissue is the pancreas, and this is spleen. Okay, and then this is one single mouse model. And then on the day of implanting this imaging window, we injected this mouse pancreatic cancer cell here. Okay, and then basically we made the orthotopic pancreatic tumor model. and then see how these tumors, this green one is the pancreatic tumor cells, how these tumors are grows over time. Let me show you another example. So this is the where we inject the pancreatic cancer cell. This is day 11 and day 18. And then let me zoom in. Okay, so this is the day 11 to day 18. So we can directly see that this how these pancreatic cancer cells infiltrate into the pancreatic tumor tissue, pancreas tissue, like this. And then we can also see how the vessels, this pancreatic tumor vessel, changed over time. Sorry. So this is the from day 11 to day 18. Again, I like to highlight this is the in vivo conditions. So there are many, cells are here. Although we are only looking at the pancreatic cancer cell and the vessel, there are many other stromal cells, immune cells, and all other microenvironmental factors are present in here. But anyway, we can see the longitudinal changes, and then so what really happened in this in vivo condition for several days to several weeks. Okay, this is finally this is the cranial imaging window technique. This is basically we do the craniotomy. So we remove the skull with a diameter of 3 millimeter, and then we put the transparent cover glass on here, and then protect the exposed skull with a dental cement. And then after this surgery, we can directly see the brain through this transparent glass. And then after recovery, this mouse can survive more than 1 year, even 2 years. So we can do very long term, the long distance imaging of the brain. So this is the one data. So through the glass, we can see the dura, subarachnoid, meninges and cortex, like this one, up to 500 micrometers. Another data. So this is GSTAX data. So we're starting from the dura and the meninges, and then we can also see that this cortex, and then distinguish the capillaries and the single cells inside the brain. Okay, and then using the cranial imaging windows, we can also do this, PK, analysis. So this one is this red color is actually antibody drugs, and then upper panel is control antibody drugs, and lower panel is modified, engineered antibody drugs to penetrate the BBB more efficiently. So this is right after 6 hour, 24 hour, and 48 hours. And as you can clearly see, this engineered antibody can more efficiently penetrate the BBB and then go into the brain, right? Like this one. Without this engineering, without this modification, normal antibody drugs cannot penetrate the BBB. So they only stay inside the blood vessel and eventually disappear, like this one. So this is another, example of the, drug delivery monitoring using imaging window. In this example, it's a cranial imaging window in the brain. Okay. And then let me just show you, explain about another technique. So by using it, we can, target the, this one single, penetrating arteriole in the brain through the cranial imaging window, like this one. So we can see, this flow was completely stopped in just a few seconds. And after that, after this, microinfarction in the penetrating arteriole, we can do, 1 month observation and then how this, brain tissue changes over time. So if you look at it, we can see the tissue expansion, basically edema, brain swelling, and then atrophies. And then, so in the acute phase, we can see this leakage of 3,000 Dalton dextran in red color. So this is same area, so same blood vessel. So we can see the leakage, so brain swelling happened at peaked at day 3, but spontaneously they recover at day 5. And then by looking at the individual cells in the brain vessel, we find out this red cell here, this is actually a pericyte, and they change their shapes very rapidly during these recovery phases, and they support the BBB function recoveries. And then this is another, 25 days longitudinal imaging. So this is same area, same vessel, same vessel, same vessel, and same vessel. And this green color is microglia. And then we can see how this microglia, changes over time, and then how they are how they behave during the this course of the micro cerebral microinfarctions. Okay, so we have this multiple sets of imaging window optimized for each organs, and then all these imaging window accessories are optimized and then make it to be used with our microscope. So if you want to image all these different organs, these imaging windows can be a great help to achieve longitudinal in vivo imagings. Okay, and then let me just go through. So we also have, as I told you, we have this heating system, so we can keep the mouse body temperature to be stable during the long-term imaging. So so you just open this sliding door, we can put this heating system. It is plate with the sliding stages. So put it in. And then also we can use gas sanitizations like isoflurane. So we have these tubes adapter on the side of our microscope box. So we can use and then, so we can we also provide these tubes and a head mask. So you can keep the mouse to be anesthetized for quite a long time using isoflurane. Okay. And then this is our standard model, but recently we also made these larger versions to handle the larger animal. So this one example. So this is rat. Okay, so we can image the rat, ferret, or even some medium-sized animal using this version. More large open space to handle this kind of live mouse, medium-sized mouse model, medium-sized animal model. Sorry. So even in the lab, we can also, obtain these several resolution images from all these different organs, like we did in the mouse model. And then we also have another version, which we call RMS model. It's we miniaturize our microscope like this one. We attach it to this articulating robot arm. So this is how it looks like. So this robotic arm has a function of human cooperations. So basically, human can move our microscope and then place it anywhere you want. And then after we lawfully place our microscope, then we can control this robotic arm in micrometer scale, and then obtain these several resolution images from the large animal like a pig, dog, or non-human primate, or even human, as you can see in these videos. Okay. Still, and then this is microscope, so we still achieve these subcellular resolutions. And then it can be good use to image the human cancer tissue during the operations. And then again, I'm gonna skip the details, but we can also develop the AI model to automatically detect the cancer nodules and cells in this tissue, in human cancer tissue, and which can be a useful tool to detect the cancer margin during the operations. And then we can also we can change our image into the different style, like conventional HN images like this one. So this two photon image can be transformed into the more conventional HN images like this one. Okay, what about the software? So although our microscope is optimized for the live animal imaging, we can also easily handle standard slide sample using adapter like this one. And then this is and then we can, so, and then our imaging software, imaging analysis software, which we call Studio, IBM Studio, is capable of handle all this large 2D imaging data, 3D data, and 4D imaging data. Okay, so let me just go in. So this is 2D large area images. Also, we can handle, organoid model. As you can see, this is one example obtained from the human lung organoid model. And then also we can handle all this 3D data, this GStack data, and then we can provide this 3D rendered data. Okay, and also this 3D and time lapse, which we call 4D data. So this is in vivo observation of the cell movement in the tissue, in this case skin. And we can see how these individual cells are moving in 3D space. Okay, so all right, so I already almost finishing my talk. So intravital microscopy is a useful tool to obtain this 3D, cellular level molecular functional images from the live animal model. We can perform this intravital microscopy in all these different organs to obtain dynamic 3D images of various to visualize the various cellular level dynamics, such as cell trafficking, cell to cell, cell to microenvironment interaction in a live animal model. Also, for drug development, we can use the intrametal microscope to monitor the drug delivery to the target cell and then monitor their efficacy after the delivery and then validate the mode of actions. Again, in a live animal model in vivo. All right. So if you're interested, please visit our webpage, ivimtech.com, or just simply send us an email through this email address, information@ivimtech.com. So in United States, we have several references. So if you're interested, if you wanna know about their feedback, please tell us. Okay. So thank you very much your attentions.
Great. Thank you, Dr. Kim.
Okay. All
So it
right.
looks like there was one question, if you can see that.
Uh-huh. Okay, in Q&A, I have one question. So, based on the applications you presented, could you clarify the mouse age at which the imaging performance is optimal? Well, mouse age doesn't be really a problem. So we can start to image the mouse as early as like 3 weeks, even 4 weeks old, um, all the way up to 1 year old, even 2 years old. So age is not the factor when we perform this kind of imaging analysis. Uh, okay, so I hope I answered your questions properly. And then next question is it possible to use other sized animal instead of mice? Yes, I can. Yes, we can. So as I showed you, we can handle, let me, okay. So as I showed you, but not just the mouse model, we can also handle land model like this, like this one. So even our standard microscope can handle mice and rat. And this modified version with a large open space, we can handle even larger, more larger animal like a rat, ferret, or even rabbit. And then even larger non-human primate, we have another options like this robotic arm systems. Okay, I think I have answered your question. Yeah,
Perfect.
if you have any further question, as I told you again, please send us an email at any time to information@ibimtech.com.
Wonderful. All right. Well, thank you, Dr. Kim, and thank you everyone
Mm-hmm.
for joining us. You have
Thank
like the
you.
links in there in the chat. Looks like there might be one
Mm-hmm.
question. Yeah, that's just thank you.
I should just thank you.
Yeah, just yeah, it looks like there's links in the chat
Mm-hmm.
for the IBIM website and also for the FLAIR, for the conference. So we hope to see you there, and
Mm-hmm.
thank you all for coming. Have a good rest
All
of your day.
right, great. Thank
Bye bye.
you. Thank you, everyone. Bye bye.